This
Morning, good
conference will now
afternoon,
be recorded.
good evening from Brenda from the Food Safety and Food Security Small Working Group on Health Commission. I hope everybody is doing well. Today we will start our first part of the webinar series of One Health Approach to Food Safety. Our first webinar is going, the title of our first webinar is about the impact of farm biosecurity and food safety and food safety. Our first speaker is Professor Cesar Gavidia. I'd like to share the floor with my colleague Amino to share the bio of Professor Caesar with the attendee. Thank you so much. Please, the floor is yours, Amino. Amino, Alamin.
Okay, can you hear me, everyone?
Yes,
Yes.
we can hear you.
All right, thank you, Randa. Hello, ladies and gentlemen. Of course, my name is Aminu. So, as Dr. Randa has mentioned, we are starting with Professor Cesar Gavidia. So, Professor Cesar is a veterinarian currently working in the School of Veterinary Medicine at Universidad Nacional Mayor de San Marcos in Lima, that's Peru. Professor Cesar is a veterinary epidemiology laboratory for more than 25 years. Prof teaches biostatistics, veterinary epidemiology, and zoonotic diseases. Prof's main area of research is public health and zoonosis, mainly parasitic infections such as cysticercosis, granulomas, Bartonella, Chagas diseases, among others. Professor Cesar has been doing field epidemiology, experimental infections in animal models, diagnostic test standardizations, animal clinical trials for testing new drugs or vaccines against those parasites in animals, etc. Professor, if you are ready, I think the floor is all yours now. Thank you.
Okay, thank you. Thank you very much for all of you. Thank you, Randa, for being a very well organizer. And you look very strong following the group every day with new messages. And I'm really happy to belong here. Thank you for the invitation. Thank you for giving me this opportunity. Thank you, Amino, for the kind presentation. Um, First of all, I would like, or second, I would like to apologize if my English is not very fluent or correct. I probably mix some words with Spanish, but I will try. I will go slow. I don't know how much time do I have, but I will try to cover some aspects related with these zoonotic parasites that has an influence, of course, in as a foodborne parasite infections in many countries. So let me know if I have some problems probably with the audio or when I'm sharing my presentation. Can you see it now? Is it correct? Can you please let me know?
Yes, yes, Professor, everything is clear. The floor
Okay.
is yours.
Okay. Thank you very much again, Randa. So as I was saying, I'm happy to belong to this group. Very interesting. The One Health social science, the One Health approach that we are using. This is for me as an epidemiologist, I'm happy to finally find the place for interact with the students, with colleagues, from different perspectives, but with just one main objective, which is One Health. And food, of course, is part of this approach of the One Health and the security and the good food that we are eating every day. So let me share with you in the next few slides two important foodborne parasitic infections. I have been working with these two of them for many years, as Aminu said in the introduction. One is the Tinea solium, and the other one is the Cainococcus granulosus, that are very, very and highly prevalent in countries like Peru, but also in many countries in South America, in Central America, in Africa, in Asia, and also in developed countries, developed nations. So let's move a little bit here. Peru is here just for you to know, and you're welcome to visit at any time. I hope you someday you could come here. And this is Peru. We are almost in the middle, in the top of the South America countries. Peru is here, and just to let you know that we are divided in three main regions. One is the coast near to the Pacific Ocean. Then we have the Andes Mountains, and this is in brown. You will see this is the highland of Peru, and then in the other part is the jungle. And why I'm saying this, because depending where you are, and even some people talk about the north, the central part, and the south, the central and the south. Depending where you are, of course, the climate, the weather, the customs, the food, the type of animals, how the people raise animals are completely different. And diseases like the ones that I'm trying to talk about this afternoon or evening for some of you, uh, depending where you are, you will find more or less infectious with these two parasites. So let's go ahead. Probably you already had lunch. I'm not. I haven't. I'm going to have lunch after this. This is a Peruvian cuisine. One of the dishes that you could find here. This is sweet potato here in that part. Of course, this is onion, lettuce, fresh lettuce, and a lot of pork, fried pork that we call chicharrón. I have seen this in other countries, but this is one of the most popular dish with made with pork in Peru. We have many dishes, of course. Here is another one. I've heard a bit, but I don't know if this is something or everything is okay. I hope everything is okay.
I guess everything is okay. Is there
Okay,
anything?
I've heard
Yeah.
some beep, but anyway, I can continue. So this is another one. Uh, with everything is made with pork. You can recognize here a lot of skin and fat. Again, this is made with pork. So we eat here in Peru and probably in many other countries, we eat a lot of pigs, a lot of pork. And why I'm presenting these dishes is not because I also cook, but this is the main source of infection for Tinea solium cysticercosis. And of course, the fresh salad, fresh vegetables, raw vegetables. Interesting is that the Food and Agriculture Organization of the United Nations, FAO, have made a ranking of foodborne parasite infections. And the ones that are in the top that you can see here is Tinea solium and Echinococcus granulosus. I mean, there are some criteria for classifying the parasites in this way, up to 24, you can see here. Probably some of you may say Toxocara should be here in that part because Toxocara is very important. But among the different parasites that are transmitted by vegetable, by fruit, by water, or meat, like in the pork, the two most important are Tinea solium and Echinococcus granulosus. Echinococcus multilocularis, if you recognize here, is the number 3, and then you can read you could have also the access of this list in this website, and I can share the presentation after with Randa. But let's focus in the first two parasites that I'm presenting in this slide. Maybe some of you remember the Tinea solium life cycle. I'm gonna show you this interactive life cycle that one student of me prepared many years ago. But I want you to refresh maybe or remember the life cycle of the parasite. I will do it the same for Echinococcus granulosus for you to see where exactly the food, the vegetables, the water can play a role in this life cycle and also in the transmission and the infection, of course, of humans and animals. So we can start here. The Taenia solium, the larval stage of the Taenia solium, we can start anywhere, but the larval stage of the Taenia solium is in pigs, it's in the pork, it's in the muscle, also the brain and other parts of the organs, but mainly in pigs is in the muscle. I mean the part that we normally consume, or the skin, or the subcutaneous. This is the larval stage. So when people eat an undercooked pork, piece of meat, or they can get infected with this. And I'm going to show you some pictures there. You can get infected with this small cyst, like a 1 centimeter, and each one of these has the potential to mature and develop into a new tinea in the small intestine. This is the famous tinea solium. In Spanish, we call it like a long tapeworm, because I suppose that the tinea solium grows alone, but sometimes you could find 2, 3, or 4 big tinnias, cestoid, of a worm in the intestine of humans. Okay, so you develop the adult stage, which is the tinea solium, and then people, I mean, normally people in the field. This is what I was trying to present in the map of Peru. In the rural area where there's no sanitation, the infrastructure for hygiene is very poor. There's no water, tap water. They have to get the water probably from river. There's no bathroom. There's no electricity. So normally people in the field go to the bathroom everywhere. So they contaminate with eggs the environment, and the pigs can reach those eggs in the environment while they are rearing around, because people don't have the corrals. The custom here is to raise the pigs outside, so pigs get their food by themselves. So the contamination is in the environment, or pigs, pigs are, how do you say the word? I forgot the word, sorry. Pigs eat human feces. They like to eat human feces, coprophagia. I was trying to remember the word. They go directly and eat the feces, the contaminated feces, with the eggs of the Taenia solium, and they get the infection directly. So what happened after that is that you close the life cycle, forget the brain and the eye there. But when the pig eat the eggs of the Taenia solium, they completed the life cycle, and again you will have a new pig infected with the larval stage, the cyst. Up to that part, someone could say, okay, this is a veterinary problem. But when the human gets the infection by eating the same Taenia solium eggs, then humans can develop a serious neurological problem, which is known as neurocysticercosis. Actually, the cyst can develop in the brain, in the central nervous system in general, in the eye, in bones, in muscle, I mean, everywhere. But mainly in humans, the eggs go to the central nervous systems. So you will have the serious problem in humans: neurocysticercosis and epilepsy. WHO has recognized epilepsy produced by Taenia solium as one of the highest problem. Actually, there is a big program in WHO trying to make an standardization on how to control Taenia solium, because epilepsy is increasing in all developing countries. So this is our friend or enemy, if you want. Let me connect my laptop. It's running out of battery. Okay. So this is a Taenia solium. It's a very long flatworm, meters long, that live in the intestine. They have hooks, they have everything, and it is attached to the intestine of the human. So human, if I go back a little bit to the life cycle, humans can be a definitive host carrying the Taenia solium adult stage, or an intermediate host carrying the cyst, the larval stage, in the brain or everywhere in the body, while the peaks are only intermediate hosts. Okay? So here you have the larval stage. This is what I'm talking about, the Cysticercus cellulosae. You will see in the left picture all of these white white dots that you are seeing here is Cysticercus cellulosae. It's a cyst, and each one of these, and you have beautiful pictures, of course, in this part on your right. Each one of these can develop into an adult stage. So each one of these can produce a new Taenia solium, an adult Taenia solium in humans. This is another picture for you to see how high could be the burden of this infection in pork. This is for a pork. It's an uncountable here number of cysts. Each one of these could produce a new Taenia solium. So people in the highlands and also in the coast, in the areas that the disease is highly prevalent, like in this picture, they consume the pigs. They consume these pigs, even though they have all the disease around, because they don't know. They don't know that this is a parasite and they can potentially be infected and they can potentially be acting as an intermediate host, developing neurocysticercosis. So you go to the highlands, or you go to this poor area where pigs are around, they they need something for the lunch. They kill one of the pigs, probably like this, and they eat it. Okay. Of course, this
Okay.
huge burden of infection is not very common to see. Normally, you will have some pigs with I don't know 100 cysts in the whole body, or maybe 1,000 cysts. But sometimes you could find pigs like with this heavy infection, thousands and thousands. There are more cysts than actually muscle here in this pig. You could find also cysts in the brain. There are many here. Also, this is a heavy infection, and the same figure you could also see, of course, in humans. Huh? Interestingly enough, is that pigs doesn't develop symptoms, neurological symptoms, evident neurological symptoms to be evaluated. While, of course, you can imagine in humans, if you have this number of cysts in the brain, you may have, for sure, you will have some problems, some neurological problems. Here you have another pictures. This is a small baby Taenia soliums that we have worked in the lab, putting the seeds with bile and did some reagents, and they are still alive. So they get out and they are ready to attach in the small intestine of a human. So what happens in humans? I already told you something. What happens in humans when they get the infection with the Taenia solium larval stage with the cyst? So here you have some pictures. There could be subcutaneous. You see another one here. The cyst can develop under the skin, and they sometimes it is palpable, so you can touch it. And sometimes, in the majority, unfortunately, the majority of cases of human cases with cysticercosis develop neurological problems because the preference of the cyst to go to the central nervous system. And here you have what happens with a patient. So you could count how many holes, how many cysts are in this brain. You have all of these are cysticercosis. You have another one here. Of course, this is a necropsy. You have even you can see the small scolex inside of each one of these. You see, here you have the cyst. So you can imagine. I'm not going to talk because I'm not specialized I'm not a neurologist, but you can imagine all the problems that someone could have if this number of cysts are present in the brain, are developed in the brain. And this is in the eye. Here's a big one, a big cyst in the eye. So I just wanted to, at this part, I just wanted to point out how important is to understand the life cycle. I call back to the life cycle, how important it is, because the transmission, again, the transmission could be for pigs directly eating the contaminated feces from humans, and in the case of humans, could be eating the pork, contaminated pork with disease and develop the neutenia, or eating eggs. Yeah. I know it's for us it's very normal. I mean, after going to the bathroom, everyone wash with soap and water the hands. But in the poor areas where there's no water, no soap, no electricity, no bathroom, no nothing, they won't they will not do that. So they can get the infection by themselves in their hands, or get the infections by drinking water, contaminated water, or by eating raw vegetables, or by eating another type of food. I'm gonna show you one of the famous cases of neurocysticercosis in a Jewish community. Okay, so let me go back to this part. Let's go back to the food, because I want to show you now the life cycle of the other interesting parasite. So again, we have fresh salad, lettuce, spinach, whatever you want to put in your salad. They might be contaminated with eggs of parasites. This is another, although I have eaten this in other countries, here in Peru, we like to eat visceras from livestock, from sheep, from cattle, from pigs, etc. And this is liver. This is one of the famous also Peruvian dishes here, eating fried liver with onion and so on. And this is another one, but with this is made with lungs that we call bofe, lungs, potato, etc. I'm not gonna give you the recipe now. Okay, so the problem here with this fresh vegetable is obvious. We can get eggs from different parasites. Let's concentrate in Taenia solium and Echinococcus granulosus. But here I'm showing this picture because the Echinococcus granulosus larval stage again is a cyst that develop mainly in liver and in lung. I'm going to show you the pictures now. And when in the slaughterhouse, the animal that goes eventually to the slaughterhouse, if the veterinarian see the cyst in the liver or in the lung, it have be get rid of the viscera. So they are confiscated, and this is a loss for the owners, because normally everyone sells the liver and the lungs and everything from the animal itself. So if this is confiscated, that means that I'm losing the money for selling these visceras. So Canococcus granulosus is the other one. It's an interesting parasite that can be transmitted also by food. You will see this in many countries. This is, of course, is the Peruvian highlands. So you have people, this combination. You have the animals, the flock, sheep, cattle, horses, goats, here South American camelids or camels, whatever you have, and then you have dogs. This combination, these three components in a special environment, makes that the parasite remain for decades, for years, for more than 100 years, for forever. It's so complicated to control not only Taenia solium, but also Echinococcus granulosus under these circumstances. I'm not pretending to change this, because people live every day with doing this, generation under the generations. So this combination of dogs, animals, and people, that creates the excellent conditions for the Echinococcus granulosus survival, and also the interaction between humans and dogs. Dogs are the pets everywhere you will see, but they are not only pets. As in the picture here, they take care of the flocks, they take care of the animals. Some of them are specialized in keeping, guards the animals, and others maybe have double function, and others just live with people in the house that you can see here on the back. But that close contact between humans and dogs increase the risk for getting the larval stage of Echinococcus granulosus and develop disease in liver and lung mainly. Could be in other organs, but those are the target organs for Echinococcus granulosus. And pictures like this, I mean, beautiful, beautiful girls, but they are starting in contact with dogs, and it has been demonstrated that if the dog has Echinococcus granulosus in the small intestine, dogs are everywhere in the dogs, from the tip of the nose up to the final of the tail, everywhere is with eggs. So this is the life cycle for you to remember, or probably you don't know. This is the life cycle of Echinococcus granulosus. I'm talking only Echinococcus granulosus. There are other Echinococcus, but we don't have in Peru. Well, we don't have. We have Echinococcus vogeli, but it's less important. It's important since there are some people infected with that, but not too many studies have been done here in Peru, at least with Echinococcus vogeli. So I'm concentrated in Echinococcus granulosus, which is the most prevalent Echinococcus everywhere in the world. The tinea is very small. It's that's the big difference with Tinea solium, which is long. This is very small, less than a centimeter in length, and eggs are eliminated and shedding in the feces of the dogs, domestic dogs. I'm talking domestic dogs, could be wolves, could be foxes, but the most important definitive host for Echinococcus granulosus is the domestic dog. So eggs go to the environment. There's a contamination, of course. Forget the humans for a minute. The pasture, the environment, the water, the plants, everything can be contaminated with eggs of Echinococcus granulosus. And with the animal, the intermediate host, sheep, cattle, horses, pigs, camelids, eat the pasture with contaminated with the eggs. It develops the disease in lungs and livers after a long period of time, usually years, several months up to years, 1, 2, 3, 4, 5 years, depending how long will live the animal. So what happens is that when people in the field find this cyst in the liver or lungs, they intentionally feed the dogs with those viscera because they don't understand, because they don't know the life cycle, they don't believe you if you try to explain how the life cycle is, etc., etc. This is unfortunately, the conditions of the rural, the isolated communities that we have here, and we have many everywhere. So they close the life cycle, and each one of these cysts, each one of these that you can see here, and I will show you another pictures, contain hundreds or thousands of larval stage of the Echinococcus granulosus. So each one of these cysts, I'm telling you this because this is important in terms of the biomass, the productivity of the Echinococcus granulosus, why it is so difficult to eliminate in an endemic area, because each one of these cysts contains thousands and thousands of protoscolices, the protoscolex, which is the larval stage, and each one of these is able to produce a new tinea in the dogs. So humans, public health, zoonosis, humans get infection here or here by playing with dogs, by getting in contact with infected dogs, or by eating pallets, by drinking contaminated water, etc. So let's move on. This is the parasite, very small, it's not very long, it doesn't produce too many eggs as Tinea solium. This is the protoscoleces, the small tinea. Let me show you here. Now we tend to use more cystic echinococcosis rather than Hairati disease. We are trying. You can see this liver and lung here. Sorry for my handwriting. So there are so many cysts. This is a heavy infection of a sheep in the Central Highlands, in the Peruvian Central Highlands. So many cysts, but apparently the animal is is healthy. I mean, you see the animal and you cannot imagine that the animal is carrying this amount of cysts and so huge. So that means this is an animal probably of 5 to 6 years. It takes a long period of time for the cyst to develop. But each one of these, this one, the one that I put in number one, can contain 10,000, 40,000, 50, put the number in thousands, number in thousands of protozoales, and each one of these could develop into a new tinea. Of course, not all of them have the ability to do that, fortunately. But if the dog eats just a piece of this, is enough, it's just enough to develop a new tinea in the small intestine. And the cycle, of course, continue. This is another one. You can see here another one, a big cyst here in the liver, sometimes only the livers, sorry, in the lung, sometimes only in the lung, sometimes in both, sometimes in kidney, heart, spleen, muscle, bones, even in the central nervous system. Some other pictures, the liquids that we have from the cyst. So what happens when the human is infected with the eggs of the Cainococcus granulosus? Here you are. I mean, this is from internet. You can download the picture. This is huge. This is an amazing number and huge cysts in the abdomen. This is from the Central Highlands of Peru. You see here almost half of the lung, one side of the lung has a big cyst, probably 15 centimeters in diameter. Here you have another one, another cyst in the lung. Some people in the highlands here in Peru, I'm talking about, can be operated because they have to go under surgery. They can be operated more than once, of course, during his or her life. Here you have in the liver, huge cyst. These two. Look at this huge cyst. This is 15 centimeters in diameter. It has like a small cyst inside a big one. That happens when the cysts have the time to develop until this size. So where are these foodborne parasite diseases? Because I'm talking about Peru, but where in the world? Here you have the world distribution of Tinea solium. The red ones, the ones that are in strong red, are the endemic or some people call hyperendemic areas. And if you remember the world map, and where is Peru in the world, is there. So we are in red, and also South America, Brazil, Colombia, Bolivia, Ecuador, we are in red. Even Central America, all the countries, Mexico, Guatemala, Africa, you see here, and Asia, all of this part, and even some countries that belongs to the developed nations. So you could find not Tinea solium, I mean, not pigs carrying the larval stage, no cysticercosis necessarily, but you could find Tinea solium in some people that migrate from endemic countries to developed nations like the US, Canada, or Europe, and they are carriers of Tinea solium, and they potentially can contaminate the food or the food for other people. I'm going to show you the famous case of this Jewish community that in New York, in the United States, that they were infected because they have a domestic worker at home that comes from endemic area, and at the time that she was serving the food or preparing the meals, they were contaminated the meals, and they developed neurocysticercosis. And if you try to remember the figure of this picture, you will see that there's a match with Echinococcus. This is Echinococcus granulosus, or cystic echinococcosis. So you see Peru, go back Peru, or you see Asia, Africa, South America, we are in the same, we have the same problem, almost the same problem with these two parasites. Of course, there are more other parasites. Probably you have the map of Toxocara, or you have the map of water availability, or you have the map or Cyclospora, another parasite, you will have the same overlap of this map. But well, unfortunately, Peru is there. We still have a lot of cases of Echinococcus granulosus or cystic echinococcosis, as well as cysticercosis or neurocysticercosis in people. There are other countries, of course, in South America that you can see here: Brazil, Argentina, Chile. They have implemented programs to control Echinococcus granulosus and cystic echinococcosis for more than 50 years. Chile, Argentina, Uruguay are working very hard, more than 50, probably 60 years. And still Peru, we are not be able to develop a national program for controlling cystic echinococcosis. Not yet, but it will take time. But you can see that after so many decades in the countries that are working to control the disease, still they are having problems with the parasite. And the same for Tinea solium, the same. And do I have? Okay. So why do we still have those parasites in our countries? Here I will show you some of the reasons or risk factors for these two parasites. The poor areas, the poorest rural areas, lack of hygiene sanitation, lack of adequate infrastructure, lack of top water, lack of electricity, custom of raising animals for surviving purposes. Of course, they have the animals for eating for themselves or for selling the animals. So in some way, this is the bank for them. They save the money by raising animals like pigs, like sheep, no? These conditions are very common in many communities. I know it's not only in Peru, it's everywhere that have the same conditions, that are still suffering the same conditions for living under these circumstances, under these characteristics. We will have problems with parasites, among other infectious diseases, of course. Raising pigs free in the environment allow pigs to go where people have been defecated. Because of the habit of the pigs that they are coprophagic, they will know and they will look for human fecal samples and they will eat it. And that's the more efficient way for pigs to get infection. Of course, could be the water. You can imagine that if someone goes near to the river for defecation, or just in an open environment, and then we have the rain, we have the river, so we disperse the eggs everywhere. You have these conditions in these areas, in the rural areas. People identify some places for throwing the trash, and also the same place will be for defecation. So pigs are very intelligent. It's a very intelligent animal. They will immediately recognize where people are defecating, where they go for the bathroom, and they will go there. You see, in some places you will have this kind of a bathroom latrine, if you wanna call it in that way, but it's very rustic, very there's not technique to do that. They just put some walls, and there's a hole probably, or there's nothing, and they just defecate inside, like this one. This is more just a few walls, if you can call walls. This is the door, and so pigs can go in anytime they want. Yeah, you can see here. I'm sorry for this disgusting probably picture, but that's the real feel, and this is one of the reasons why we are still have cysticercosis or Tinea solium in our communities. Not one of the reasons. I have mentioned many reasons around this picture that you are seeing here. Also, like probably in other countries, we have this commercialization of different type of animals, and there's no control basically here. There's no control, veterinary control. I mean, there's no veterinary control. You cannot cancel also this. It happens during the week or particularly during the weekends. People bring animals, people purchase animals. They just disperse the different infectious diseases. Let's talk about Taenia solium everywhere. They can get an animal there, go to another place, or they can buy an animal, an infected animal, even if they know that the pig is infected. They will kill it and they will cook it. So the transmission, the life cycle, if you remember, the life cycle will continue forever and ever. It's so hard to control this, so hard to change this this scenario, these circumstances that are around. I mean, it's not only it's, we don't need to look at the disease only from our veterinary medical perspective, but also there's a big social component, there's an economic component, there's I mean, there are so many factors around, educational component, of course, but it's so hard to control. I'm not trying to be pessimistic in controlling or eliminating diseases like this, but we have to work very hard to do that. This is just a picture of the another community. As I was telling you before, this is not only a problem for low-income countries, but also in developed nations. And this is the example I was telling you, the seroprevalence of cysticercosis in an Orthodox Jewish community. So there was I forget the numbers, I can share all of this with you, but there was a seroprevalence in this community, and I suppose they don't eat pork. So they, after doing the analysis, the epidemiological analysis of these positive people, the results showed that they probably get the infection through the domestic worker at home who came from endemic countries in Central America. And this is the most famous case. There were also four people developing neurocysticercosis. Some of them with epilepsy. They were definitely positive to the test. You can see the ages. I mean, it's 6 years, 16, 34, 39. And in the epidemiological analysis also demonstrated that the transmission came through someone who works at home, domestic work, that was originally from one of the country in Central America, the endemic area for Taenia solium. So we can consider that, of course, the most prevalent area are our countries, but also people that are going to other countries in the world can be developed or can be considered as a transporter of the infections. Talking about again Echinococcus granulosus, but could be for also Taenia solium, it was very interesting to me to find in this community in the Central Highlands. This is the health center in Tambochaca. For those who might understand Spanish, and they have a drawing here in the wall and the entrance of the life cycle of Echinococcus granulosus. Very interesting. Someone painted that drawing, and they identified that the disease can be transmitted by eating vegetables. But it was more interesting to me that I was asking people if they understand what is painted here in this drawing, and no one can identify what is in there. So it's like education is important. I agree with that, of course. Education has to be for everyone, but it's not enough if you paint or if you make a propaganda or you make some small book for kids. It's not enough just once or a year. You have to continue with education for years and years. And this is why education has failed so many times trying to control diseases, because the program has to remain for too many years. It's so difficult to teach, it's so difficult to change the behavior of killing the animals or eating pork contaminated. It's so difficult to do that. Changing behavior in people if someone here works is an anthropologist or sociologist, may understand better than me, but I have seen this in the community. So hard for us to make them understand the life cycle or explain what we are doing with the parasite. Again, this is the factors that, just to show you the risk factor for developing and maintaining the life cycle of these two parasites as an example. And of course, this one, people, this is a sheep. People need to eat or need to sell some animals, and they kill it at home. And you see, even cat. There's a cat here, but you can see one dog, two dogs, waiting for the owner to give them the viscera as a reward. They consider that if the dogs eat the viscera, raw viscera, they get more courage. They're more brave to take care of their animals. So everything will be for them to feed. You will see these pictures, these kind of pictures there. They are killing another sheep, and there's a dog waiting for a reward for being feeding. The contact with the, I already showed you these two pictures, the contact with the dogs, and also I cannot call it an abattoir. It's just a place for people to kill the animals and commercialize them. This is another one in the south, in Peru. It's completely open. They kill sheep and they slaughter sheep, and also South American camelids, alpacas, llamas, and you will see dogs are always around. That's another one. I guess I'm running out of time, so. And this is in the north, so the disease is everywhere here in Peru, mostly in the central highlands and in the north. There's a place that people, instead of sheep, they have goats because of the mainland is completely different. But when a goat die, they just leave it they just through the body everywhere or wherever, um, and the dogs are around and they are eating, but they starting eating the viscera. So these parasites have studied. Sorry to say that, but it's like the Taenia solium and the Echinococcus granulosus have already studied parasitology before us, and they have adapted. Of course not, but they have adapted perfectly to these kind of circumstances, the environment, the behavior of the people, and also the behavior of the animals, either the intermediate host or the definitive host. So this is very interesting. If you think, sometimes I think like if I were a Taenia solium or if I were a Echinococcus granulosus, what do I need to survive? And then you will find very interesting all these small components creating a perfect scenario, perfect thing for surviving of the parasite. Again, furious. So finally, maybe some of you might have this question like me. Are there parasite eggs in food? Of course, we have. We have parasites in water. We have parasites in vegetables. We have, I already showed you, parasites in meat, like in the pork. Um, there could be other parasites, of course, but just Taenia solium. And we have parasites eggs? Yes, we have. There are The next question could be, aren't there many studies, several studies that demonstrated that the food or the vegetables or water are carrying parasite eggs? Not too many, unfortunately. So there are more, if you enter in PubMed or Google or whatever you want to search, you will find more studies in humans, more studies, several studies in pigs, several studies in dogs and humans talking about Echinococcus granulosus, but you could not find many studies, as much as many studies as the ones that I mentioned. So there are not too many studies demonstrated eggs of Echinococcus or eggs of Actinia solium either in water or vegetables particularly. But there are some, there's a few of them, like this one. And I apologize, I mean the table probably you will see is so small, but here almost at the end you will see Echinococcus granulosus. This study was, I guess it was in Sweden or Switzerland. Oh yeah, I can see. It was not particularly for humans, but they bought vegetables and fruits that were used for feeding captive primates. But this is the same market that people can get the fruits and vegetables for them. And they demonstrated that along other parasites, there was Echinococcus granulosus in two of them. Here you have. No? Here you have another one. This was they were evaluating first a methodology to look for eggs, parasite eggs in vegetables and also water. I think so. But they finally they did this sampling. They obtained 157 lettuce samples and they evaluated the eggs by PCR, of course, and now everyone is using by this advanced technology. And they collected samples from farmer markets and also from supermarkets in Switzerland. This was in Switzerland. What they demonstrated at the end that they found, let me see here, they have Echinococcus multilocularis, which is very close to Echinococcus granulosus. The life cycle is very similar, but the parasite requires a fox, so they have foxes in that area, and you can see there are from farmer markets, sorry, from farmer markets, they found two samples that with contaminated with eggs from Echinococcus multilocularis, while in the supermarket they didn't find anything. But that's important because this is Switzerland. So I was wondering when I saw this paper, I was wondering what could be happen here in my country, in Peru, or in yours. What could happen there or here where there's not too many restrictions or too many cares for this kind of vegetables like lettuce or like spinach or whatever other vegetable you know. So this is a contamination with eggs of Echinococcus multilocularis. This paper also, although they don't demonstrate the eggs, they make an interview, and in the interview, one of the risk factors they found important was that eating raw meat or vegetable was associated with a coproantigen for cysticercosis, for Tinea solium. And you can see here the odds ratio is very high. They didn't demonstrate it by finding the eggs in the food, but just for the interview, they say okay that could be also a risk factor. Forget this. There's a cloudy table, of course. I just wanted to point out that this is the studies. This is a systematic review of the studies trying to demonstrate tinea eggs in vegetables and fruit. So this is a compilation of many works that have been done so far. I don't remember this paper was published. I think it's fresh. Yeah, last year was published last year. So there are not too many studies demonstrated eggs, but everyone says that the contamination could be through or the infection could come through water, fruit, vegetables, meat, or just dirty hands. When you serve or someone served you the meat or the cook or the meal in generally, and the dirty hands, like in what happens probably with the Jewish or Orthodox community, the contamination could come there. I'm finishing with this. I guess I have only two more slides. Again, you see, this was a study in France, I guess. Yes, it was in France. Again, you see here rural vegetable gardens. Rural vegetable gardens were sampled, and they found here in dogs, in cats, and in total, they have some positive samples in the soil. So there's a contamination with eggs of this is Echinococcus multilocularis again. Yeah, we don't have here in Peru Echinococcus multilocularis, but I'm interested. I mean, I'm in contact with some of these researchers. This one here, Alvarez Rojas, who is now working in Chile, to make a study here in Peru with vegetables and also probably water. Let's see what happens. And finally, besides the vegetables, the water. There could be other ways of transmission, and probably yes, this is an area that haven't been explored too much, which is the houseflies and other insects, other vectors that can carry with them some of the eggs of the tinea. For instance, for Tinea solium, we have demonstrated one of the dung beetles. I don't remember the scientific name. One of the dung beetles that that play with the human feces can eat the eggs of the Tinea solium, and then if a pig eats not the feces, eat the dung beetle, they can get the infection, so they can develop the cysticercosis, the larval stage. And again, this is sorry for these tables also. This is just to show you that this is another systematic review of the importance of the vectors, the transmission the parasite through vectors, through different vectors. There are some coleoptera, there are some diptera here for tinea, for different teneas, and you can see, if you don't see it, don't worry. There's one study that demonstrated Tinea solium with Coleoptera, and in the table which is down, in table 2, again Coleopteras and Dipteras, and then you have Multilocularis and Echinococcus granulosus transmitted through these kind of vectors. So there is not only water or raw vegetables or undercooked meat, but also vectors like this that are everywhere. So maybe your dish is clean, and you have some fly there with the eggs carrying to different places, and then you can get the infection. Of course, the likelihood might be low. Nobody knows exactly, because this is an area, as I said before, that need to be studied deeply. Okay, that's it to me. This is just picture from our highlands. I mean, I don't want you to be scared of coming to Peru. You're welcome to come anytime and enjoy, of course, our country and our cuisine. And just keep the hygiene, and that's it. You can eat this underground. This is cooked underground. Guinea pigs, chicken, pork, cattle, everything you want to put there, fava beans, and then you enjoy the lunch. Thank you very much. This is my family years ago. Thank you, Randa. I don't know if there are some questions.
Oh, gracias, Professor Caesar, for your nice presentation. Actually, that was an impressive presentation, especially that part that you discussed the hydatid cyst and the numbers inside of the animals. We appreciate that a lot. There's a lot of details, and it was a simple at the same time. Thank you so much for your great presentation. And now the floor for the audience. In case anyone would like to ask any question, please mute yourself and ask Professor Pete Caesar. If you wish, you can send him an email. We will share with you his email. If there's any private or more research related questions you want to ask it to Professor Caesar, you can email him also. So the
Yes.
floor is, yeah, thank you. So the floor is yours, our great audience. Thank you.
I hope you could understand my English. I'm sorry if I stop
No,
sometimes.
no,
Um,
yeah, yeah, that was great.
Listening.
It was clear English. Yeah, please, Joseph. Joseph or Michael?
Hello.
Hello, good evening, good afternoon.
Hi.
Hello,
Muchas
Michael.
gracias for the presentation. And
Thank you,
really,
Michael.
I even have a headache after all that information and all the depressing information we got from about this highly successful parasites. But I wanted to ask if you have encountered any moderate to successful control program in any region of your country? Because I'm asking in the context of having seen that any top-down approach is really not sustainable, because like you said, there are a lot of factors that affect the success of any program, given the economic and other disparities of the communities that live with these challenges. But even if that's the case, is there any light in the tunnel or any concerning control programs?
Yeah, thank you, Michael, for the question. That very important question. Yes, I wanted you to see that there are many factors around when you want to think in control these kind of parasites, probably for other infectious diseases as well. Yes, there are some strategies that have demonstrated to be effective. Like let's talking about Tinea solium. There's a vaccine in pigs, very effective vaccine, almost 100% effective vaccine. But the problem with the vaccine is that, of course, there's a cost. And who is going to pay for the cost of the vaccine? You don't need to vaccinate the pigs just once. You need to vaccinate every certain period of time. So there's a cost for the program. And thinking again in rural communities and poor people, I will ask again this, who is going to pay that? If this is just, let me put it in dollars, if this is going to cost like a $1 or half a dollar, and who is going to pay for that? Besides that, people are not accustomed to the vaccination of pigs, so they will not allow you probably to vaccinate so frequently the pigs, or once a year, or every 6 months, depending how you want to plan to use the vaccine. So there's a complication in that way. There's also a very good test for detecting uh, tinea solium carriers, I mean adult stages, humans, but you have to collect feces. Sometimes you have to collect blood from humans, and again, you have to face that other problem that people don't like to be sampled every year, or every 6 months, or every 3 months, to see if they get the infection. Education is another one I mentioned, but it will take years and years and decades for people to change or be educated to control the disease. I mean, you compare the pictures I have shown you in the rural community with the cities that where probably we are living now. What is the difference? What is the big difference between these two communities or cities? The difference is just the advanced technology or the commodities that we have. I have electricity, I have water. I cannot raise pigs here in my neighborhood because people will kick me off. No, so development of the cities or development of the communities will bring will cause that the infectious diseases in general will disappear, but that change is very hard to obtain. So it's probably we have to integrate so many strategies at the same time. um, some people said, okay, you cannot allow to kill your pigs or your sheep at home. Yes, but how can people will do them? They have to pay to go to the slaughterhouse, which is far away from there. Then, if the pigs or the sheep has a cyst, they will be confiscated. So there's a loss. Who will pay? The owner, the small peasant, the owner that have only 2 sheep and 3 pigs. Who will pay them for these confiscated organs, or for the confiscation of the whole animal if this is a pig? So this so many factors around. This is why I said there's a social driven things, economical things, educational things, and also the technology and our research that we are working on. We know that we have the vaccine, we have the treatment, an efficient treatment for sheep or for pigs, depending on the parasite. We have the efficient diagnostic tests for humans and for animals. But this is for research. How do we implement all of these strategies and methodologies into a real control program? it takes long. The examples are clear. I mean, here in Argentina, Uruguay, Chile, more than 50 years working on that. Cyprus, that have controlled the disease years ago, after 100 years of controlling the disease, they have they are, the peaks of the number of the cases of human echinococcosis is now increasing. So that means that once they stop applying the control measures, the parasite was still there, probably eggs, probably one animal, and the number of cases are now bouncing up again, are increasing. So that means that you cannot um, you cannot feel that you win the fight until this is really the problem with this kind of parasites. I don't know if I answer your question, Michael.
Certainly. Thank you so much. Thank you.
You are very welcome. Is there any other question or
Hello.
it's not? Just
Yeah, so mine is not really a question, but just a comment. From the experience we have had in Kenya, especially with the Tinea solium in pigs in western part of Kenya. Previously, we used to have quite a number of epilepsy cases being reported, but among the control measures that was instituted include, people in western Kenya have quite a number of roaming pigs. Pigs are just
yes.
roaming freely. And so one of the things that we did was actually encouraging people to have a toilet, so that now, because the only means through which it can spread is through fecal contamination more often. So the number of cases had really gone down because of that control measures. And then another thing for cystic echinococcosis, we also have it in some parts of northern Kenya, which is where it is endemic. And because livestock have been moving from those endemic areas to new areas, we are now getting new cases of cystic echinococcosis moving from known areas to new areas. And then cases are now increasing because people are not really aware of the disease in these new areas.
Yeah.
That was just a comment. Thank you.
Yes, important. Yeah, important comments. Thank you, Joseph. Yes, that's the reality. I mean, I always say this: it's very easy to write down a control program in a Word document, let's say, and put everything that ideally we have to do this to control the disease. We can understand that, but once you want to put it in the field, then you have to be aware you have to face with all of these difficulties, as you mentioned for cystic echinococcosis. And the other one for Tinea solium that you are controlling. I'm glad to hear that. But this is so difficult for people in some communities to say that they have to tie or they have to make a corral, build a corral for pigs. Pigs destroy the corral that they prepare with the materials there. I mean, poles, these kind of things. Pig will destroy everything. You have to construct a big wall for pigs. But that means that they have to. I see this in one community, in many communities here in Peru. Once the pigs are corral, someone has to be in charge of bringing water or bringing food to the pigs. This is why people leave the pigs outside, roaming freely everywhere, because they get their food by themselves. Nobody takes care of them. And just they call the pigs during the evening or the night just to keep them inside the house. But otherwise, if they have to corral the pigs, they have extra works to do. This is why sometimes they don't want to do it. For cystic echinococcosis, there was another measure that people say we have to boil the viscera before giving the dogs the viscera, before feeding the dog with the viscera. We have to boil it. That means that you have to get something for the fire. You have to put a big pot with the viscera, wait until the water is boiled, and remember that this is in the altitude, 3,000, 4,000, 5,000 meters above sea level, so it takes time for boiling. You have to put the viscera, wait for that, wait for the viscera and the water is a little bit cold for feeding the dog. That means someone has to be in charge of that. And usually you don't have someone extra in the family to take care of, to be in charge of all these extra activities. So there are so many problems around. But of course, measures, control measures work. Yes, of course they work. The thing is how we implemented these measures, these strategies in the field where the parasites are present. That's the problem. That's the key issue, and that would be a challenge, of course, for all of us. Thank you, Joseph.
Oh, thank you so much, Joseph. Thank you, Professor Caesar, for the clarification. Also, there's Miss Hamid. She would like to ask a question. And I apologize, she's going to be the last person before starting her presentation because we are running out of time. Thank you so much.
Hello, can you hear me?
Yes, we can hear you, Miss Hamid.
Okay. Thank you very much, Per, for the presentation. Please, I would like to know if there is a simple test, like home tests, that one can do before symptoms are produced. In the case of Tenia solium,
I didn't hear you well, but I guess you were asking me for a test that can be
home-based
used,
test.
home-based test that can be used for the
Yes,
diagnosis of tinea solium
yes.
adult stage or the larval stage. I mean, neurocysticercosis or tymiasis.
At the early stage, that's the larval stage.
Larval, okay, I can tell you both. I mean, we can use screening. There are some screening tests or serological tests for the larval stage. The most famous one is a Western blot test that detect 7 specific bands for cysticercosis in general. For the larval stage, it doesn't matter if this is in the muscle or the brain or any other part of the body. It is very good. It's very sensitive, highly sensitive, very specific test. There are some ELISAs. There's another test that also can be used depending the availability of the laboratory. For teniasis, the simple one is not specific. I mean, detecting X coprological test detecting X of tenias doesn't mean that this is tinea solium. Could be tinea saginata, could be Diphyllobotrium, could be any other tinea. So there are some specific PCR and very sensitive PCR tests for detecting tinea solium X in humans. But again, if you want to use it for a screening, you have to take samples of everyone and do the PCR and see if some of them will be positive. The problem with the adult stage of tinea solium, as I said before, that the adaptation of the parasite is very interesting. So tinea solium requires only 1 to 3%, 4% of people with adult stage of the parasite to remain stable and endemic in an area. So let's say in another words, if you take a screening test for 100 people, fecal samples, run the PCR, you should expect to find only 2 or 3 of them positive to tinea solium. So yeah, but this is the way that you can make the diagnosis. Some people here are also using for research. For research, they make a screening and a coproparasitological test, the common one, and then the positive people, positive to X, tinea X, they receive how do you say, a purge. They drink something for purge the parasite, and they collect the whole fecal sample, and then they identify the proglottids are different between tinea saginata and tinea solium. That's another way to do it. But yeah, the problem is how to do it in the area and convince people to bring the samples. Is that clear? I don't know, Miss Pupola.
Um, she has mentioned in the chat box, Professor, that
Oh,
thank you, Professor. I mean, home-based, like using strip, which are easy and affordable, like the case with malaria test strip and gonococcal test kit, for
ah,
example. She just mentioned.
oh, I don't know. I guess for Tiniasolium, I don't know if there's a test for using like with the strip. I guess no. I guess no. But probably I could be wrong, of course. I'm not working too much in the diagnostic test for tinea solium. But I haven't seen any, even for echinococcus. I haven't seen any test for dogs. In that case, for the definitive host. I haven't seen for echinococcus and also for tinea solium a test like this. That would be wonderful. I'm pretty sure that there's any of these tests like this. It will be very useful because we don't need to go to the field, which is 8-10 hours from the city, and go back with the samples for running in the laboratory. That's the ideal thing. You collect the sample, you make the evaluation with the strip, and then if the person, or the dog, in the case of echinococcus, is positive, and you immediately can give the treatment. Yeah, this is a place. This is an open. This is a question for the research. Yeah.
Yeah. So, thank you
Mhm.
everybody for your valuable questions to Professor Caesar. Also, thank you so much, Professor, for your answers. In case anyone he has or she has any questions to Professor Caesar, we will share your his email. Or please, Professor, you can just type it in the chat box in case anyone would like to ask your question. Before moving on to our next session with Miss Bukola Hamid. So let's have 2 minutes break, 3 minutes drinking water, just refreshing ourselves, and we can start our new session. Thank you so much, Professor Caesar, for your
Thank you.
great presentation. That was awesome. You're awesome. Thank you so much. Thank you.
Thank you. Thank you, Randa. Thank you all. Thank you. Thank you very much.
So I need to go to drink some water. I'm coming back, okay? So welcome back everybody to after 2 minutes break. Actually, that was an awesome presentation. Thank God it's recorded so everybody can benefit from that valuable presentation and info. And now, not from the veterinary field, from another field, but it's part of the One Health. As well as we have noticed from Professor Caesar that One Health and the part of discussing that zoonotic diseases, parasitic zoonotic diseases, it leads to the importance of health equity, uh, and that is all about One Health. So today also we have another presenter, Miss Bukola Hamid. Are you with us? Hello. Uh, I can't see her name, but she has asked the questions to Professor Caesar. Can you hear me, everybody?
Yes, I can hear you. I think maybe she has went to drink to have a drink or something.
Okay, so we can wait her 3 minutes more?
All right.
Yeah. So
This
welcome back,
conference will now be
everybody.
recorded.
So welcome back, everybody, after just listening to Professor C's session. And now we are resuming our program for today of the impact of farm biosecurity. And now our next session is going to present Ms. Bakula Hamid. Ms. Bakula Hamid, she holds a bachelor degree and a master's degree in microbiology from Kawara State University. I hope that I spell it correctly, from Nigeria. She was a graduate assistant in the same institute, the Kwara State University in Nigeria, and currently she's working as a freelance writer. So welcome, Ms. Bukala Hamid, to our session
Thank
today.
you.
It's our pleasure, yes, to have you with us, to be a potential speaker. So now the floor is yours. Let me check. Can you yeah, enable?
Yes, I can see the screen.
Great. So let me just a minute. The slide. Okay, so the floor is yours, and the impact of farm biosecurity and food safety. Thank you, Ms. Hamid Bukala.
Thank you very much for having me. I'm very sorry. I'm not familiar with the GoTo app. So as much as I would have loved to share my slide because I've made some changes, but it's okay. Thank you all. Good evening from Nigeria. I'm Bukola Ahmed. And like she rightly said, I was a graduate assistant at Kwara State University, and currently I'm a freelance writer. I'll be talking on the impact of farm biosecurity on food safety. Please, can I have the next slide? Now, farm biosecurity are set of measures or practices which stop the spread of diseases on the farm. So these processes are put in place to curtail the spread of infection On the other hand, we have food safety, which is the process by which foods are handled or stored to prevent infection. Now, why are all these in place? The essence of this is to make sure that we eat. The essence of farm biosecurity is to ensure that we eat safe food. Please, can I have the next slide? Okay. So now, farm biosecurity comprises various measures, like I earlier said, all in a bid to prevent diseases or hazard. And these hazards can arise from microbial origin, be it parasite, like the professor had earlier mentioned, be it viruses, be it bacteria, be it zoonotic origin, that is diseases transmitted from humans to man and vice versa. It could also arise from genetically modified organisms, as well as microbial products such as toxins. Now, what are the measures that can be put in place to curtail this hazard? One, we have containment controls that restrict movement between infected and non-infected areas. That is, whenever there are visitors coming to our farm, you have to put measures in place to make sure that all these infections coming from the visitor doesn't get to the farm animal, or the farm animals transmitting those infections within the animals to the visitors, and then it gets to the environment. Another measure is to create boundaries, management of disease boundaries. This is done by introducing traffic control, which I will later discuss rather during my presentation. Also, we have socio-technical interventions such as testing. We also have vaccination of wild animals. This prevents them from different diseases, like the professor said in his own in his presentation. One of the means to prevent Taenia solium from getting to humans is to vaccinate the pigs or sheep, as the case may be. So that's vaccination. Vaccination is also a measure in farm biosecurity. Then cleaning and disinfect, that is cleansing of equipment, cleansing of even the farmyard itself, and then disinfecting using various disinfectants for the cleaning of those equipment. Also, we have risk management. This is also the same thing as the disease boundary control, in which case measures such as vaccination, testing, cleansing are put in place. Please, can I have this next slide? Please, can I have the next slide? Okay.
Yes.
Now, what are the key
Yeah,
components or the key measures of farm biosecurity? We have one, sanitation. Sanitation, that is the cleaning, is just a basic hygiene. Just like we humans, we have to take our bath every day, brush our teeth. The farm also has to be in a very good hygienic condition. So that is a good hygiene practices on the farm is necessary to maintain a disease-free herd. Now, isolation. When new animals are brought to the herd, there's a need for them to be isolated from the old ones, so that if there is any infection, they will be quickly treated before they are introduced into the herd. Then traffic control. This traffic control serves two purposes. One is to inform the road user of an incident. For example, if there's an outbreak on the farm, there are certain traffic measures traffic signs that can be put in place to show that, oh, there is an incident on the farm. I would have loved to show this pictorially. I've done that in my new slide, but unfortunately I can't share my slide. Now, the second reason for putting traffic control measure in place is to make sure that vehicles as well as visitors coming into the farm are directed to the right place. So all these measures are put in place to this may involve movement to a screening area. That is, the traffic control measures are put in place to ensure that both visitors and vehicles go to the appropriate area, such as the screening area or a cleaning and disinfecting area in case of visitors, they may have to go to screening to ensure that whatever materials they are bringing into the farm are not infected. Then at other times, when new animals are brought into the farm, they may need to go through routine check before they are introduced into the herd. And at other times, when equipment are brought into the farm, they may need to be cleaned or disinfected, as the case may be, before they are introduced into the farm. Please, can I have the next slide? This is still on key component of farm biosecurity. Another key component of farm biosecurity is resistance. Now, this resistance can be obtained through vaccination. When animals are vaccinated against particular disease or infection. Hello, can everybody hear me? They develop immunity that makes them resistant to the same infection. For example, in case of Taenia solium, if the pig is vaccinated against Taenia, the likelihood of developing the infection then becomes very reduced, if at all it occurs. Then the last point, aside from resistance, if I'm correct, should be security. Hello? Hello?
Yes, hello.
Okay, that's assessment. Okay, assessment before security. Okay, the assessment has to do with going through all of those farm routines, that is the isolation, the sanitation, traffic control. This measure is taken to ensure that all the other measures earlier stated are quite in place. That is, like I said, there are different components of farm biosecurity. We have sanitation, we have isolation, we have resistance and traffic control. So what assessments as a component of farm biosecurity does is to ensure that those measures are all in place. Please, can I have the next slide?
Yes. Yeah, here it is.
Please, can I? Okay, now I'll be talking on the importance of farm biosecurity. So why is it important for these measures to be put in place? One, it helps in risk management. In the incident of an outbreak in the farm, when the right biosecurity measure is in place, this disease can be easily curtailed so that it doesn't spread within the herd or even from the animal to humans. Maybe the humans taking care of the animals, or even humans coming in as visitors to the farm. Then it also results in an enhanced productivity and sustainability. For example, with the right farm biosecurity in place, the animals are less prone to diseases. And with that, if the animals are being bred for, say, milk production, their milk production capacity will increase, because in the absence of disease, the animals are prone to produce more milk. So in which case it eventually increases productivity of the animal, and also sustainability, in the sense that once the farm biosecurity measures are in place and intact, you can always go back to those measures and say, oh, these measures are what I used earlier to increase my productivity. Okay, that means they are accurate to maintain my farm, and then you can go on with it. Then profitability, of course, when the animals are producing well, once animals are producing well, you increase your profits. Your profit eventually increases. Then we have food security. Now this is general. The general importance of farm biosecurity. Once the right farm biosecurity measures are put in place, there won't be any fear of food scarcity, because be it an animal farm or a plant farm, once the farm is free of diseases, the productivity of the plants and the animal tend to increase, thereby leading to food security. Then policy formulation. One importance of farm biosecurity is it helps in policy formulation. For example, if you look at the earlier lecture by the prof, Taenia, for example, why it's still so rampant in Peru is because there are no policies made in place to tell to or inform farmers as to, especially the local farmers, pig, as to how to keep their environment, as to measures to put in place to prevent the Taenia solium, the egg and the larva, or the last stage of it that spreads to human. There are no, those measures are not in place, and these measures have to do with the farmer themselves. So once farm biosecurity measure practices are put in place, it helps in policy formulation, so such that one can, as a body, now, policy can be made that, oh, if you are rearing pigs, these are things you should put in place in your farm. For example, vaccination. For example, isolate new piglets or pigs that are coming into the farm. But for local farmers, since they are not practicing be made. Then we have human and environmental health. Again, I will still use the prof's lecture as an example. See, the environment.
Hello, Miss Hamid, are you with us? Hello. Hello, everybody. Can you hear me?
Yes, I can hear you. I think she's muted.
Oh my God. I hate. Yes. How can I unmute her? Miss Bukola, you are muted.
Maybe you should send a mute request.
That's the problem. What kind of application? I don't know. It's not Zoom. I can't unmute her. Keep muted, pin. Okay, okay, send unmute request.
Miss Bukola, can you please unmute yourself?
Yes, please, Miss Bukola. Because oh, sometimes. And only 6 minutes left to the meeting.
Yeah.
Okay. Miss Bukola, we can't hear you because you are muted.
Maybe you can try typing the problem in the chat box so that we can
Yes.
see you.
Oh my God. Oh my God. Okay. Uh, Bukola is unmute yourself. Can you see my screen?
نعم
So,
أنكاوتن
can you hear? No, we can't hear you, Miss Bukola.
This conference will now be recorded.
Oh,
This
I'm going to
conference will
resume the
now be
recording.
recorded.
And
Can
please,
you
Miss
hear me now?
Bukola, now
Can you
we
hear
can
me
hear
now?
you. Yes, yes, we can hear you, Miss Bukola. So now we will
Okay, well,
go
I'm
back
at the
to
end of my presentation already.
yeah, great. So yeah, so
So
yeah, yeah.
I'll just briefly go through the impact of, farm biosecurity on food safety. So generally, farms with good biosecurity practices have a better control over diseases and pathogen spread in the food chain. So once pathogen spread and diseases have been curtailed in the food chain, one can say that the impact of farm biosecurity on food safety is a positive one, like I had earlier stated in the importance of farm biosecurity. So I believe the problem with food security and food safety is arising from lack of these farm biosecurity practices, especially with the local farmers. So those farmers who breed say pigs or cow on a local scale or home scale, they don't put up all of these practices. They don't they tend not to vaccinate their animals. They tend not to be hygienic with both the equipment and even in breeding of the animals. They tend not to put certain practices or such as isolation. Once they get a new animal, they just introduce it into the herd. They don't test, they don't do any routine test to see maybe there's any animal infected. So once farms put in place biosecurity measures, we can be sure of taking safe food. So I'll end on the note that farm biosecurity practices are indeed important to have safe food. Thank you very much for listening.
Thank
Hello?
you so much. Yeah, we can hear you, Miss Hamid. Thank you so much for
Okay.
your for your great presentation about the impact of farm biosecurity on food safety. You have
Thank you
summarized
for
the
having
essential
me.
points. Yeah, no, you're welcome. You have summarized the essential points regarding that, and now the floor is for the audience. In case they want to ask any question to Miss Hamid, please go ahead. The floor is yours before the session ends. And thank you. So, is there any questions from the audience to Ms. Bukola Hamid? In case you have any question, please go ahead, or you can send her an email. I'm gonna type down. I'm gonna just copy paste her email into the chat box, and you can email her. Okay. So again, I would like to thank everybody for attending today's webinar. It was our pleasure for hosting this webinar. We are the Food Safety and Food Security Small Working Group from the One Health Commission, and that's me, Randa. I would like to thank everybody for being part of this webinar today. Special thanks for our presenters, Professor Caesar. He is also part of our team, and Miss Bukola Hamid for their nice and great presentations. So before ending our session, I would like to wish for everybody a good day, a good afternoon, or a good night. Thank you so much. Have a nice day or evening. Bye bye.
Thank you so much, Profs. Thank you, Miss Hamid. I hope everyone will have a nice day. I hope everyone has shared his Gmail.
Yes,
Okay.
yes, in the chat box. Yeah, I'm gonna share them with you, Amino. Don't worry, we can download the chatting, okay?
All right.
So, yeah, thank you so much, everybody, for your time today. Have a lovely day or evening. Bye bye. Good night. Good day. Bye bye. Thank you, Professor Marcella.